I very much appreciate the opportunity to come back here to Grand Hotel, and thank you to Naventus for the audience. And this is a glorious moment because the first time I was on stage at Grand Hotel, I was actually the moderator, like you, Katarina. And the presenter was Professor Olle Hernell, presented Lipum, and he was looking for seed financing. Already at that time, I was fascinated by the approach and the medical need, and I still am. And in 2016, I became the CEO of Lipum, and since then, we have actually been successful with the seed financing. So we have raised around about 18 million euro, and we did an IPO at First North in April, 157 days ago. So we still have around about 5 million euro on the account. So we are not looking for financing today. And more we want to report what's going on. And it's very nice then to report that Professor Olle Hernell was just recently awarded, because of his research in the field of pediatrics and the human milk, breastfeeding, because that was in research in that area, he discovered the target protein that we are working with today in Lipum. We have also recently reported that we progress. We are halfway in our tox studies of our lead candidate. And since the tox studies are progressing very well, we have initiated the GMP manufacturing of our lead candidate, which is a monoclonal antibody. But we work with chronic inflammatory diseases, and inflammation is a natural process. The problem arises when the inflammation is lingering and puts the patient in a constant condition of inflammation. And the most well-known among people is rheumatoid arthritis, because it affects around about 1% of the global population. Another type of arthritis is the juvenile idiopathic arthritis that affects children. And both of these diseases have been or are of special focus for Lipum. So we have developed a monoclonal therapeutic antibody, and it's called SOL-116. It is directed towards a protein called bile salt stimulated lipase, BSSL, and it was this protein that was discovered by the founders of Lipum. And it turns out that this protein is an overlooked target molecule in the inflammatory process. So the preclinical work has been focused on first of all, validate the target molecule BSSL as a target. The other part of the preclinical work has to be validating antibodies to block BSSL and to get the therapeutic effect, that is, to stop the inflammation. And for that reason, there are, in the preclinical field, there are ex vivo studies, so there are numerous of ex vivo studies, what you do in a tube or something like that, but there are also in vivo studies on test animals. And this has been validated in a number of different in vivo models. And this has led also to approved patent for treatment of rheumatoid arthritis, juvenile arthritis, and pending on inflammatory bowel disease by blocking BSSL. And that was approved 2010, so it's still valid for another 10 years approximately. But more recently, we have applied for patent for the antibody SOL-116, and that will give us a protection for our product until 2040. But there are also other opportunities for regulatory protection by orphan drug designation or by pediatric investigational plan or special support you can get from the authorities for biologicals. And this is, of course, something that we will pursue. An example of an in vivo study is pristane-induced arthritis model, where you expose rats to pristane. And the control rats are getting serious ill, they get arthritis, while those animals that get an anti-BSSL antibody are significantly less affected. And this data you see here are on experimental antibodies, but this has also been validated with our lead candidate, SOL-116. However, the SOL-116 is a humanized antibody, so it has considerably less affinity for the BSSL that the rats have. This is also, in the filled dots here, represent also two different ways of administration of the antibody, which show us that we can use subcutaneous injection of the antibody that will be just as good as interperitoneal injection. And we have been mostly focusing on rheumatoid arthritis and juvenile idiopathic arthritis. Firstly, for validation of the target molecule. Secondly, because there is a high medical need in the juvenile idiopathic arthritis, and there is also very good models to validate both the target molecule and our lead candidate. But on top of that, we also have data for other indications. And we believe that BSSL as a target has importance in numerous of different chronic inflammatory diseases. We can see data, we have ex vivo data for other diseases, but we also have in vivo data on so-called knockout mice. That means mice that don't have the BSSL molecule in the circulation. And there we can see that for arthritis, actually the mice that don't have a BSSL in the circulation, they're completely unaffected, and they don't develop arthritis at all. When it comes to IBD, inflammatory bowel disease, or in this specific case, ulcerative colitis, we can see that the mice that don't have the BSSL are significantly less affected compared to the control mice. So we have good reasons to believe that our approach and that SOL-116 is a drug candidate that can be applied on many different diseases. However, we focus on a disease called juvenile idiopathic arthritis. It's a rare disease affecting children up to around about 16 years old. It's much more common among girls than boys. And the children, beside the pain and direct effects of the disease, there are also significant social consequences, because fatty dew leads to that they don't want to play with the friends or go to school. The parents have to stay home, etc. So the social consequences are large. And juvenile idiopathic arthritis bear the same problems as rheumatoid arthritis. And that is that after the initial treatment with chemical drugs like methotrexate, if that isn't sufficient, the patient gets biological drugs, and most commonly a TNF inhibitor drug, like Humira from AbbVie. But with the children and the adults, around about 30% of the patients are non-responders. And an additional 30% are actually patients that stop using the drug because of the side effects or loss of efficacy. So then they need an alternative. And it's important that that the treatment is going on with the children, because there's a very high risk that they develop uveitis, that is inflammation in the eye. So when you look into this group of patients, so even that this is a rare disease, there is a more common rare disease, because around about 600,000 children in the Western world have the juvenile idiopathic arthritis. But if you look into the fractions of patients that actually are not helped with today's drug, you end up with a market opportunity to up to 1.2 billion euro. And we think that this is something that we can address from Lipum. And we also intend then to apply for an orphan drug designation, in particularly in the USA, where we see best opportunity for getting an ODD. We also have reasons, based on the preclinical data, we also have reasons to believe that SOL-116 can be combined with other therapeutics, maybe even with other biologicals, because data suggests that SOL-116 will not be immunosuppressant like the other biological drugs used today. And in fact, if you look into the drugs that today are in phase 3 studies for juvenile idiopathic arthritis, all of them are drugs that are used for rheumatoid arthritis, having these drawbacks with non-respondence and significant side effects. So there is no really new and unique approach to treat either rheumatoid arthritis or juvenile arthritis patients. And we believe that with the less immunosuppressant drug, we can have SOL-116 to be potentially a drug that is combined with some of the other drugs on the market. So if we look then on our development plan, this is something that we have been communicating also in connection to the IPO. Our main focus has been to validate SOL-116 in tox studies. We are half through them now. To produce under GMP that has been started. And this is all aiming for a clinical trial application during the spring next year. And then we should start clinical trials phase 1 on healthy volunteers round about mid next year. And that is the phase 1A study. But we also plan for a phase 1B study, and that will involve adult RA patients. And the idea is then not only to monitor the safety, but also to monitor pro-inflammatory biomarkers to get a first ID on the therapeutic effect of SOL-116 in these patients. Then later on, in the next step, we proceed in the phase 2 study, we proceed in a study that will be a mix of adult patients before you enter into children with juvenile idiopathic arthritis. But very important for us is that we, in parallel with this, also expand our preclinical data on more indications and on the mode of action. And altogether, we think that we will have a very attractive portfolio of data on patients and in the preclinical work that will attract interest from partners for co-development, and in particular, there is a possibility to run parallel phase 1B studies on different indications. You should also keep in mind that according to the statistics, biological drugs that have entered into phase 1 studies have twice as high chance to get FDA approval compared to a small molecule. And if you look into the space of autoimmune diseases, it's actually 4 times higher probability for an FDA approval. So there is, of course, an opportunity for deals in a rather early stage, and there are examples, quite recent examples of deals made also already in the preclinical stage, but in particular then in the phase 1 stage. So this is obviously something we will pursue if there is opportunity, but we focus on finding someone that wants to explore the parallel indications in parallel to juvenile idiopathic arthritis. So, who are doing this work then? Yeah, well, when I saw this stage, I was happy that I'm a former handball player, because you could easily go and fall over and you get too excited. But that's, of course, obviously not the reason why I'm managing Lipum. But this is a team, and we are fighting. And we are 4 full-time employees managing the company, and we have almost, some more than this photo actually, that are consultants with retainers that work more or less as employees with the same engagement and enthusiasm for the goal that we have set up for Lipum. And if you look into the board of directors, there has been changes. We have 2 new members just recently, and the board of directors is transforming from being mostly scientific oriented. Now it becomes more business oriented, and this is something that is likely to proceed further on. And we have a very nice and engaged board of advisors with experience both in the clinical side, in the autoimmune field, in rheumatoid arthritis and juvenile idiopathic arthritis, but also on the more scientific side in the inflammatory field. So to wrap up, the press releases recently told that we have, after the 4 weeks dose-finding studies in the tox program, we concluded that we could actually see no negative effect from SOL-116. Then we could start the GLP compliant study that we need for our clinical trial application, and that is moving on right now in 1 species. Once this was concluded, it made sense to start the relatively expensive GMP manufacturing of SOL-116, and that is made in 500-liter scale, which means that it will provide us with SOL-116 for the clinical program, including also phase 2 studies. We are fine-tuning the clinical development plan, everything with the aim to start phase 1 studies mid-next year. Then along this journey, then we will apply for an orphan drug designation, and that will then focus on juvenile idiopathic arthritis. Just as important is then to exploit the value of our lead candidate, and that is to study more indications, and that is our focus in the preclinical work. We look into, in particular then, interest in inflammatory bowel disease. But there are some others we are looking into as well, according to the list I presented earlier. And in the frame of this, we have a strategic collaboration with Örebro University, and that is partly because it's supported also by the Foundation for Strategic Research, but also because they have very interesting models there for atherosclerosis, and this is also related to a heart attack, and in some aspects also to cancer. So we are expecting very interesting results from that collaboration. And then I wanted to highlight Immunid. It's a company in Uppsala. They have interesting models for looking into what's going on in the blood, and that helps us to find out more about the mode of action. So if I wrap up, why invest then in Lipum? Well, we work in a field where there is a very high medical need. We have identified a completely new and unique approach by having this new target, BSSL. We have a fully developed lead candidate, SOL-116, and all this is for a market that is gigantic. Thank you for your attention.
Thank you very much, Einar. To look a little bit even further ahead, what are your expectations for Lipum, let's say in a 5-year perspective?
Sorry. Yeah, absolutely. According to our long-term plan, I mean, a lot of focus in a company like Lipum has been to validate, because we introduce a new target molecule. We have to prove to others that it makes sense, this target molecule makes sense. So a lot of focus has been to validate the target. Then the next step is to validate the lead candidate, that it works on the target. But once you have a lead candidate, the SOL-116, as we saw, it's much higher probability to get an FDA approval with a biological drug. So then we need to exploit the value of our product, which means that we will continue to focus on studying more indications, both larger and smaller indications, but most of these indications are anyway very large market-wise. And by getting more preclinical data for SOB116, we will show partners the potential. And if we then have tox data finalized, we have phase 1 data finalized, then all these checkboxes are checked for them, so they don't need to wait for results from safety studies. And altogether then it will make us very attractive for collaborations and all kind of deals at that stage. So I think we will have partnering with some companies on other indications in the 5 years' time, definitely. But also on our main track, what we're doing ourselves, we have submitted and I get an approved orphan drug designation. And if everything best comes to best, we also have an FDA approval for juvenile idiopathic arthritis.
You spoke earlier a little bit about collaboration. How about competition, specifically in the field that you're now focusing on, specifically on the SOL116?
Yeah, well, as you saw, the juvenile idiopathic arthritis is a market of 4 billion euro. Rheumatoid arthritis is a market of 24 billion euro. And the indications of special interest for us are around about 700. So, of course, there is competition. But if you look into juvenile idiopathic arthritis and look into the drugs that are in late clinical stage, they are all identical drugs that are used for rheumatoid arthritis for adults. And you can expect exactly the same limitations when applied in children as you have in adults.
The 30% that you mentioned.
So there will still be a medical need for those that are not helped with the conventional drugs.
Well, best of luck, and thank you very much for joining us today.
Thank you.
Thank you.